The overall goal of this method is to isolate trypanosomes expressing a particular variant surface glycoprotein, or VSG, from the rest of the population, and identify parasites that have switched from the starting dominant VSG to any other VSG.
This method can help answer key questions in trypanosome biology, such as what are the factors or events that influence VSG switching in a population of parasites.
The main advantage to this technique is that it's faster than previously published methods and allows for easy quantification of switched and non-switched cells.
When the lab first started working on antigenic variation, the first problem was how to detect rare switchers. This kind of problem had actually been solved in immunology a number of years ago through the use of magnetic activated cell sorting to enrich cells that were negative for a particular trait. Porting that selection process to antigenic variation seemed like a really promising idea.
To begin, Lister 427 strain bloodstream trypanosomes should be grown to a density of 0.5 to 1 million per milliliter. Aliquot a 50 × 10 to the 6 cells sample into a 50 milliliter tube, and then centrifuge. Save 1 × 10 to the 6 cells in culture to be used later as positive and negative antibody controls. Pipette off most of the supernatant, leaving roughly 750 microliters in the tube. Transfer the cells to a 1.5 milliliter centrifuge tube. Finally, centrifuge the cells at 4 degrees Celsius for 4 minutes at 2,800 × G. And then